Hello, good morning and good afternoon. My name is Sam and I am a Global Product Manager here at QIAGEN. In today's webinar, I wanted to talk about improved DNA amplification techniques for using downstream analysis such as next-gen sequencing, microarrays, and PCR. In today's webinar, we're going to focus on using improved DNA amplification with our newly released REPLI-g Advanced DNA single-cell kits. To use these, the DNA that we amplify from a select number of cells for human karyomapping in order to phase single-cell gene defects. I need to remind everyone before we get further into our presentation that the QIAGEN products shown here are intended for molecular biology applications. These products are not intended for the diagnosis, prevention, or treatment of a disease. For up-to-date licensing information and product-specific disclaimers, feel free to look at the QIAGEN kit handbooks or the user manuals. These kit handbooks and user manuals are available at QIAGEN.com. They can be requested from QIAGEN Technical Services or from your local distributor. So in today's webinar, we're going to focus on the process of amplifying limited amounts of genomic DNA from eukaryotic cells in order to do karyomapping. Karyomapping is often done when we have couples with a family history of a genetic disorder and they want to know whether or not they're going to have a child that could have a genetic disorder that either they are carriers for or that they have in their family. With karyomapping, this is often used for pre-implementation genetic diagnosis, such as or PGD, of single gene disorders. Down here in this bottom slide, we have an example of what karyomapping is looking for. With karyomapping, often we look at the SNPs across both the mother, father, and a reference sample in order to look at whether or not the embryo or the pre-implemented embryo is going to have a genetic disorder. So with karyomapping, we do genome-wide SNP analysis of the parents and a close relative. Typically, we're analyzing anywhere from 300,000 or more SNPs, and this mostly applies to a very limited number of cells, which means that we need to have the ability to have enough DNA to go through and use these methods. This representative haplochart is showing you the father, mother, and several embryos, and you can see the makeup of the genetic material in the different embryos, and you can see that we can have allelic representation from either one or more of the father's and mother's chromosomes. One of the issues with karyomapping is that we are performing this on a limited number of cells. So we need to collect the cells and we need to be able to amplify that genomic DNA in order to use our analysis methods. When we're looking at the amount of DNA that we're starting with, it's often a limited couple of picograms. And for doing this analysis, we may have to get up to nanograms or even a microgram, depending on the technique that we're using. Whole genome amplification can be used to overcome this limitation. And really, the final data that we get from a karyomapping analysis is really dependent on the performance of the whole genome amplification, as this is essential to the quality of the data. You can imagine that as we go through and we talk about these really important applications, we need to have kits that allow us to go through and to definitely amplify the DNA, make enough of the DNA, and then to amplify it faithfully so we don't introduce any abnormal changes. One of the reasons why we launched our REPLI-g Advanced DNA Single Cell Kit is to tackle these issues. And what I want to present to you is some of the ideas behind how we built this kit. The REPLI-g Advanced DNA Single Cell Kit works with our existing technologies that we've built into our REPLI-g product line, namely our MDA or multiple displacement amplification. The way that MDA works is it's an isothermal reaction that takes place at typically around 30 degrees Celsius. This slide goes through the basic mechanism of how the MDA reaction will work. First, we denature the genomic DNA, and then primers, which are often randomers, are going to bind to the DNA to allow us to prime the 529 polymerase to start the amplification. As the primers are extended at 30 degrees, the polymerase moves along the genomic DNA, or if we're starting with RNA, we can make a cDNA copy, displacing the complementary strand, which then becomes the template itself. In contrast to other amplification methods like PCR, our MDA does not require different temperatures. And in the end, this results in very long fragments with low mutation rates. The 529 polymerase that we use at QIAGEN has about 1,000-fold more fidelity than most of the polymerases that are used for PCR amplification. And the fragments that we can get can be very long, anywhere from 2 to 70 kilobases with these very low mutation rates. So the product of this MDA amplification can be fragmented for short read sequencing. It can be used directly for PCR analysis, or it can also be used for long read sequencing. QIAGEN has a number of REPLI-g amplification kits. And when we started to look at addressing the need around karyomapping, as well as several other single cell applications, we decided to move in a different direction and improve one of our existing kits. So we looked at our REPLI-g single cell kit, and we decided to make some improvements and enhancements on it. When talking to customers, of course, they were looking for improved quality. So what we are looking for is reproducible performance, meaning that when we start with a sample, we get enough and a reproducible amount of product that comes out of this. We're looking for high uniformity of amplification and minimal technical bias, and this is basically measured by having no allelic dropout. Customers are always looking for shorter workflows, so we simplify this down to a single tube workflow with only a couple of different pipetting steps. So the protocol that we're going to talk about is currently a lot faster than our REPLI-g DNA single cell kit, and it's easy to handle, meaning that there's only a few steps that you have to do in order to get your product. And it has a very flexible workflow. One of the things that we've incorporated into our REPLI-g Advanced kit is a single cell storage buffer. This means that you can collect the cells that you're using, put them in our cryopreservative buffer, freeze them away, and then pull the cells out when you want to process them all at one time. This sample storage buffer allows cells to be collected and shipped without having an impact on the cell quality. It also preserves the quality of the cell, which preserves the genomic DNA within the nucleus, keeping the genomic DNA from being nicked or damaged, which greatly helps with preserving the quality of the data and minimizing allelic dropout. So our REPLI-g Advanced DNA single cell kit is an improved version of our REPLI-g single cell kit. This kit has been optimized for eukaryotic cells. And the way that we're able to achieve higher quality, meaning higher uniformity and lower technical bias, is by implementing our storage buffer, our isothermal lysis that we use, and improved composition of our reaction mixes with a much faster protocol. The MDA reaction now only takes 2 hours versus the 6 or 8 hours we used to use. We've currently launched 3 different kits. We have a 24 reaction kit and a 96 reaction kit. Within these reaction kits, we have the single cell cryo protect buffer as well as all the reagents. And then we also offer the REPLI-g single cell cryo protect buffer as another standalone product for researchers that will be collecting cells, storing them, and maybe shipping them to another facility to do the amplification and the karyomapping. This slide shows us the workflow for a collection and amplification of a cell from the beginning to the final REPLI-g reaction. We collect the cell in 4 microliters of our REPLI-g advanced storage buffer. We add 3 microliters of our denaturization solution, and then we allow that to sit around for 10 minutes at room temperature. Following the lysis and denaturization of the genomic DNA, we add 3 microliters of the stop solution. We mix, and then we add our REPLI-g Advanced Single Cell Master Mix, which contains the polymerase and the amplification reaction. We allow this to go for 2 hours at 30 degrees Celsius, stop the reaction at 65 degrees for 3 minutes, and then at the end of this, we have our amplified DNA. From a single cell, we typically get 25 to 35 micrograms of our amplified genomic DNA. From start to finish, there's only 2 times that we have to open our tube in order to add reagents, and the total time is 2 hours and 20 minutes with only 10 minutes of hands-on time. One of the metrics that researchers are always interested in when they look at amplifying genomic DNA is the consistency of the results. Here I'm showing you the results of collecting single cells using one of our single cell collection tools called the QIASeq. We collected cells from the Jurkat, Lovo, MCF7, HeLa cell lines. We collected several different cells here that you can see from the number of single cells analyzed. We did this over a couple of different experiments with different operators. You can see here that the average yield when starting with a single cell was anywhere from 25 up to almost 40 micrograms of DNA with a very low coefficient of variation. Reagent lot over here refers to different reagents or lot sizes of our REPLI Advanced DNA kits, showing you that as we manufacture different lots, we get consistent yield, at least enough yield to go through and do microarray and NGS analysis. We also went through and used genomic DNA as a standard. So here we see a similar experiment where we have several different technicians going through and starting with 1 nanogram of genomic DNA, different lots of our REPLI-g Advanced kits. And you can see the mean yield that we get here is somewhere between 35 to 45 micrograms of amplified genomic DNA. This next slide shows the uniformity of using the REPLI-g Advanced DNA single cell kit. And in these instances, we're starting with single cells that were isolated using a single cell collection device. And we're comparing here our REPLI-g Advanced, which is our new kit, versus our traditional REPLI-g original kits. With the single cells for the REPLI-g Advanced, the cells were collected and put into the cryopreservative buffer. With the REPLI-g original, they were collected and put into PBS and then processed according to the protocol of each of those kits. The genomic DNA was used as a standard, and our downstream analysis of the amplified genomic DNA is a targeted QIAseq targeted DNA panel. This panel has about 240 different genes and covers about 850 kilobases. You can see here what we're looking at is the uniformity, or the percentages of the bases with a certain percentage or within a certain binning of the mean read length. Using 40 nanograms of genomic DNA or 40 nanograms of the amplified material, we've looked at the binning of 5% mean read length, greater than 10%, 20%, and greater than 30% of the mean read depth. Here you can see that the REPLI-g Advanced DNA single cell kit, shown in blue, performs better than our traditional REPLI-g DNA kit when looking at the amplified bases and the uniformity across the NGS assay. Another way to look at this is the Gini coefficient. The Gini coefficient allows us to look at the uniformity. The smaller the Gini coefficient, the more uniform the amplification is of a single sample. So here we're looking at the, in the light blue, a new REPLI-g Advanced single cell DNA kit. In the dark blue, our classical REPLI-g kit. And in red here, we're looking at the genomic DNA. And you can see here the lower Gini coefficient associated with the REPLI-g Advanced kit, showing higher uniformity across the samples that were processed. We also compared the multiple displacement amplification technique versus other techniques such as picoplex and Malbec. Here we're looking at the percent uniformity across 5%, 10, 20%, and 30% of the mean read depth. And we can see here that in the dark blue is the genomic DNA control. In light blue, we have the REPLI-g advanced kit. In the red, we have the picoplex, and in the grayish, we have the results from the Malbec. We used the same QIAseq targeted DNA panel that covers 850 kilobases, and we used each of the protocols as they are laid out in the handbook. Starting with the single cells, we can see that the REPLI-g advanced DNA single cell kit has higher uniformity. And we can see this also when we go through and look at the Gini coefficient, which is showing us the uniformity across the individual samples. In conclusion, the new REPLI-g single cell kit shows higher uniformity than other sorts of technologies for amplifying genomic DNA. In this experiment, we're looking at the allelic dropout. The way that we've looked at the allelic dropout is to use the genomic DNA as a reference. And we're looking at single allelic dropout of heterozygous SNPs. We defined a loci with a frequency of a SNP allele between 25 to 75% as heterozygous. And what we went through is we analyzed the number of heterozygous SNPs in genomic DNA and whether or not they turn to homozygous SNPs after whole genome amplification. A change from a heterozygous to a homozygous SNP would be an indication of allelic dropout. On the y-axis here, we're looking at the percentage of heterozygous SNPs with single allelic dropout. The genomic DNA being the reference has, of course, no allelic dropout. With the REPLI-g advanced, we can see we have somewhere between about 15 to 30% allelic dropout. With the picoplex and the Malbec technologies for amplifying the DNA, you can see we have much higher allelic dropout. So in conclusion, the REPLI-g advanced DNA single cell kits show a lower allelic dropout rate than other sorts of technologies. And we attribute this to a couple of different factors. The first factor is the preservation of the cell in the cryoprotect buffer, which protects the genomic DNA. The second is the isothermal lysis that we have. And the third is the multiple displacement amplification with the Phi29 polymerase. All of these things added up together lead to lower allelic dropouts. We looked at several different applications, and what I'm going to show you is some data for karyomapping that was done in collaboration with Miloslav Hornak at REPLI Biology Park in the Czech Republic. In this experiment, biopsies, which contain a few cells, were isolated from the trophal ectoderm of embryos using a fine needle glass needle, and they were transferred into PCR tubes. So the workflow is to take the biopsy. The biopsied cells, which are a few cells, are washed 3 times in droplets of the cryoprotective buffer and 0.1% PVP. The 0.1% PVP is to keep the cells from adhering to the PCR tubes. They are then, after the wash, put into 1 microliter of the CBP with 0.1% PVP, and then it is 20 degrees. Following the storage, they're processed by adding 3 microliters of the pure CBP. They are then subjected to the whole genome amplification using the REPLI-g advanced DNA single cell kit, and then they're karyomapped using the human karyomap 12 DNA analysis kit, which is a microarray platform from Illumina. Through this study, there were 10 different cases that were analyzed, and I'll show you data from a couple of these cases. With these 10 different cases, you can see the number of samples that were used. The code for these is the TE are trophal ectoderm samples, E are embryos, and IVF are the number of times that in vitro fertilization was used in the study. The disease that was looked at is shown here, as well as the gene or the genetic defect that we're looking for in the study. Across these 10 different samples, this process was used. They were analyzed on the Illumina microarray. And one of the nice things of the microarray platform is that it will give you some sort of QC metrics of the whole genome amplified DNA. The selected QC metrics that are used to judge whether or not the DNA has been amplified faithfully is the overall call rate of 85 to 99%, the AB rate of 20 to 30%, allelic dropout rate is 0 to 80%, and miscall rate of less than 5%. So what we have down here is a table of the 10 different experiments that were done. In this table, we have the fathers, mothers, the embryo in E, and R would be the reference sample, if there was a reference sample. We're looking at the call rate here. So in order for these samples to be further analyzed, the call rate needs to be above 85%. And if you look across the call rate here of experiment 1, case 2, case 3, 4, 5, etc., you can see the call rates are all very high, at close to 94 to 99%. The AB rate should be 20 to 30%. And you can see the AB rate is all within the standard specifications. And the allelic dropout rate should be less than 80%. And you can see that the allelic dropout rate here is at most around 5% in case number 1. Miscall rates also are very good at 0% for all of the cases that were studied. With the QC metrics showing very good qualifications, these samples were further processed and looked at downstream in the case studies. First case study we'll go through is called case study 1, Fragile X syndrome. This is a case where a child of 12 months old showed delayed development and mild mental retardation. The child was tested for the Prader-Willi-Angelman syndromes, and they came back negative. And then a test for Fragile X syndrome revealed 266 CGGN repeats, which is the full mutation spectrum for this particular disease. When doing karyomapping of the mother showed 111 CGGN repeats, which is the pre-mutation spectrum without a phenotype. If the couple decided to have any future children, this pre-mutation spectrum could convert over to a full mutation spectrum, and there's a big risk here of that occurring again. So in the second pregnancy that this couple wanted to have, they wanted to avoid the risk for Fragile X syndrome by using pre-implementation genetic diagnosis. So 8 separate embryos were developed. And down here, we're looking at the haplomap of the different embryos. You have the mother's haplomap. There's 2 alleles, M1 and M2. If a haplotype is linked with the mutation, it's going to show up with the star here. So the M1 has a star, the M2 does not. And then for the father, since this is X-linked, the father has 1 X chromosome, 1 Y chromosome. If he donates an X chromosome, it'll be shown here as a P. If he donates a Y, it'll be shown here as a 0. In addition to also getting the haplotype around FMR1, other chromosomal abnormalities can also be discovered by going through and looking at the data. In the end, 2 embryos were identified, 1 male, 1 female, which had the M2 haplotype with either the father's X or Y chromosome without any additional chromosomal abnormalities. And one of these was chosen for in vitro fertilization and implementation back into the mother for a second child. The second case study we have is nephrophthisis. In this particular case study, a couple with 1 healthy child decided to go through and have IVF in virtual fertilization and implementation because they were unable to have a second child. The fetus showed polycystonic kidneys under regular ultrasound check. DNA testing of the fetus from amniocytes showed that there was a homozygous mutation in the TMEM67 gene, which is the causative gene for kidney polycytosis and nephrocentesis. And so going back and looking at the haplomap of the mother and father, it showed that the mother and father are healthy carriers of the mutations in the heterozygous state. So the couple decided to perform another round of IVF using PGD and karyomapping. So down here we can see that there were 9 different embryos. The haplotype here of the mother and father are shown. Since this is no longer an X-linked abnormality, you can see that there's 2 different alleles for the father, P1 and P2. The haplotype that is linked to the mutation is going to be shown here as a star, and it's highlighted here in blue. So out of these 9 different embryos, we had 1 here that had the blue, which would not be used for further implementation. So out of these 9, we had 8 left. And then when looking at other sorts of chromosomal abnormalities, there was 1 that were detected. So this allowed the couple to go through and to pick a good embryo for implementation that would allow it to not have the TMEM67 mutation, and they selected one that was did not have a mutation in either of the alleles. So in conclusion, the REPLI-g Advanced DNA single-cell kits are highly suitable for human karyomapping. They provide reliable and reproducible results for phasing and haplotyping of single gene defects. In addition to using these with microarrays, we've also used the same kits for next-gen sequencing analysis using low-pass sequencing, as well as PCR interrogation of specific mutations. And when using karyomapping for PGDs, this helps avoid the risk of severe genetic disorders from inherited parents with a known family history of genetic disorders, and also detects chromosomal aneuploidies if present in the embryo. So in addition to be able to look at particular types of mutations or particular types of haplomaps, we can also see chromosomal number abnormalities. So with that, I'd like to thank you for your attention and coming to today's presentation. Today we talked about the REPLI-g Advanced DNA single cell kits, which are available from QIAGEN. If you're interested in or you have any questions, feel free to reach out to QIAGEN by emailing QIAwebinars@QIAGEN.com, or feel free to contact QIAGEN at this email address or at this website down here. So again, I'd like to thank you for your time and have a good day.